Welcome back to the deep dive. Today, we are undertaking a mission to really master one of the most fundamental yet dynamic fields in modern medicine, the science of viruses and the complex strategies we use to prevent and treat them.
Right.
And this isn't just about feeling informed. This is about building a foundational understanding, a mastery of the core biology, the latest diagnostic tools, and all the clinical implications, especially for pathologies affecting the head and neck.
That's the objective, exactly.
Yeah.
The source material we have today is, well, it's dense, it's precise.
Yeah.
And it really requires an elevated level of rigor. So whether you're prepping for a medical board exam or you're just, insanely curious about how infectious disease works,
Right.
you need to understand the structural differences between a nucleocapsid and a lipid envelope.
Yeah.
And critically, the difference between viral evasion strategies like antigenic drift and antigenic shift.
Because these concepts are the bedrock of everything. Modern infection control, targeted antiviral treatment, all of it.
All of it. It's the foundation.
Our sources lay out a pretty comprehensive analysis. We're starting with the acellular nature of viruses, moving through their life cycles, and then diving deep into their crucial involvement in human illness.
And we're going to be looking particularly closely at respiratory pathogens, head and neck manifestations,
Things like acute otitis media, sudden hearing loss.
and even the dark world of viral malignancies.
So our mission today is to analyze this material with the exact precision required by an elite professional. Let's unpack this, starting with the most basic building blocks.
Let's define our subject. The sources are absolutely clear. A virus is an acellular infectious agent.
That's the key thing to grasp right at the start. It is not a cell.
It's not a cell and it can't reproduce on its own. It's what we call an obligate replicator, meaning it absolutely requires a living host cell to do anything, especially to make more copies of itself.
And that acellular nature, it immediately dictates how we have to approach treatment and diagnosis. The scale difference is just as important too.
Oh, absolutely. When we talk about viruses, we are talking about structures that are orders of magnitude smaller than bacteria.
I mean, it's hard to even picture.
It is. Their size varies, from maybe 20 nanometers up to a giant 500 nanometers.
Yeah.
But the typical size is around 100 nanometers in diameter,
So compared to a bacterium,
roughly 10 times smaller.
which explains why for the longest time we had so much trouble even identifying them.
Of course, they're completely invisible to a standard light microscope. You need the power of an electron microscope or, the precision of modern molecular techniques to even know they're there.
And yet the structure itself it's elegantly simple.
It really is. Every virus is made of genetic material, a nucleic acid, and that's contained within a protective protein capsule,
The nucleocapsid.
the nucleic acid, or sometimes you'll hear it called the nucleoprotein. Same concept.
Let's use the analysis of the influenza structure from our sources. It's a perfect example to really dissect these components.
It's a great model. So we look inside. First, you find the blueprint, the ribonucleic acid, or RNA. That's the genetic material.
Okay.
And wrapped directly around that RNA is the nucleoprotein, or MP. Its job is just to stabilize and organize that genetic material.
And then providing the internal structure, the framework for the whole particle, that's the matrix protein, M.
Right. The M protein sits just under the surface envelope, and it's critical for maintaining the shape of the virion and helping with the assembly process later on.
But the parts that are most fascinating, at least from an immune and infectious standpoint, are those surface components.
The spikes.
The spikes. In the influenza diagram, we see hemagglutinin, H, and neuraminidase, N. These are the keys to the kingdom, aren't they?
They are. Hemagglutinin, the H, is what typically binds to the host cell receptors to get the whole infection started. Neuraminidase, the N, does the opposite. It's vital for cutting the virus loose from the host cell during release.
So it doesn't get stuck to the cell it just came from.
Exactly. It ensures the new particle can go off and infect something else.
And for influenza, we have to talk about that protective outer layer, the lipid envelope. Not all viruses have one.
No, but for those that do, like herpes and influenza, it dramatically changes how they get in and out of cells.
So how does a virus actually get this envelope?
It's a masterful act of biological thievery. We call it budding.
Budding.
The envelope is a lipid bilayer vesicle that the virus literally acquires from the host cell membrane as the new particle exits. It essentially wraps itself in a piece of the host.
But it's not just a neutral cloak. It's redecorated for the next mission.
Precisely. During this budding process, the virus makes sure that the host's Normal membrane proteins are replaced with its own viral proteins, those H and N spikes, for example.
Oh,
So these new viral proteins are what let it get into the next host cell. It uses the host's material, but it controls the final product. It's an incredibly potent strategy.
that's brilliant. So when researchers classify a new virus, what are the sort of 3 non-negotiable parameters they use?
It boils down to 3 structural and genetic traits. First, the type of nucleic acid is the genome DNA or RNA.
Okay.
Second, the shape and size of the capsid, that protein shell. And third, the presence or absence of a lipid envelope. Those 3 give you the full framework.
Let's go back to the viral proteins for a second, because they're split into 2 really important groups, structural and non-structural.
Right. The structural proteins are the architects. They literally form the physical parts of the mature virus particle, the virion.
So the capsid, the H and N spikes, the matrix protein,
All of those, yes.
and the non-structural proteins.
Those are the machinery. They're the essential enzymes needed for the infection cycle to work, but they don't get packaged into the final released particle. They're the behind-the-scenes workers.
Like integrase.
Integrase is a key example. It helps retroviruses stitch their DNA into the host genome. And, of course, the most famous one, reverse transcriptase, which defines the whole retrovirus family.
Which brings us to the 4 fundamental categories of viral genomes. This is where the complexity really starts, and understanding this tells you how the virus will replicate.
Absolutely. We categorize them strictly by their genetic material. You've got double-stranded DNA, or dsDNA,
Right.
single-stranded DNA, ssDNA, double-stranded RNA, dsRNA,
Right.
and the most common, single-stranded RNA, or ssRNA.
The dsDNA viruses, like viruses, they usually have a smaller amount of genetic information, just a single molecule.
Correct. And then you have the dsRNA viruses, like reoviruses. What's critical to know about them is that their genome is often segmented.
Segmented, meaning it's in pieces.
It's split into several separate fragments. And that segmentation allows for rapid change through recombination, which is, as we'll see, incredibly important when we talk about influenza.
But ssRNA is the category we see most often clinically. And here we hit that absolutely crucial distinction: polarity. Positive strand versus negative strand RNA.
This is where that elite professional understanding really begins. If a virus has positive strand RNA, its genome can be used directly as a messenger RNA template.
So the host's own machinery can read it immediately.
The host's ribosomes recognize it instantly, and protein synthesis can begin right away. It's like arriving with the final readable script already in hand.
It just starts making proteins, no conversion needed.
Exactly. The negative strand RNA is the opposite. It cannot be directly translated. It has to be converted into a complementary positive strand RNA first, before the host machinery can make any sense of it.
And that extra step has a massive implication for the virus itself, doesn't it?
Yes, that is the key clinical takeaway. Because the host cell doesn't have the machinery to convert negative RNA to positive RNA.
The virus has to bring its own.
It has to be hyper-prepared. It has to carry its own polymerase enzyme, usually an RNA-dependent RNA polymerase, right inside the virion, ready to go the second it gets inside the cell.
Which makes that polymerase a prime target for antiviral drugs.
A highly specific prime target.
So we've covered the structure and the genome. Now let's walk through the life cycle. The sources say that while they all vary, the 6 common steps are critical because they're the targets for our therapies.
Right. The process always starts with a moment of recognition. Attachment.
The key in the lock.
The key in the lock. Viral surface proteins, like that hemagglutinin, have to bind to very specific receptor molecules on the host cell membrane.
And this perfect match explains viral specificity.
Absolutely. This receptor binding mechanism is what defines tropism, which cells and tissues the virus can infect. If a cell doesn't express the right receptor, the virus simply can't get in.
Which is why a cold virus doesn't typically infect, say, bone cells.
Precisely. This requirement dictates the entire pathology of the virus.
Okay, so once attachment is secure, the virus moves to entry.
Entry is usually achieved through endocytosis, where the host cell membrane actually pinches off and brings the virus inside in a little bubble, a vesicle. Then once it's inside, the next crucial phase is uncoating.
This sounds like where it's most vulnerable.
It often is. Uncoating is the removal of that protective capsid, which exposes the fragile genetic material. This is often triggered by pH changes inside that vesicle or by the action of viral or cellular proteases.
Enzymes that break down proteins.
Right. So if we can block those proteases, we can stop the infection right there.
And the moment that genome is exposed, the factory opens. Expression and replication.
This is the takeover. First, viral proteins, those non-structural enzymes, are expressed. Then, the genetic material is rapidly replicated using a mix of hijacked host machinery and those specific viral enzymes. It's exponential multiplication.
And once you have enough parts, you move to assembly.
New virus particles are built, packaging the genetic material into the nucleocapsid, getting all the structural proteins in place. The final step is release.
And as we said, the release method depends on the structure.
Correct. Non-enveloped viruses, like rhinoviruses, typically get out via host cell lysis. They just burst the cell open. Enveloped viruses, like influenza, exit by budding, which lets them steal that lipid envelope and keeps the host cell intact, at least for a little while longer. Every single one of these steps is a potential specific target for a drug.
The life cycle shows us what the virus does. The next step is how modern medicine catches it in the act. Let's talk diagnostics. Historically, if you had a cold, the doctor just said to rest. But now, precise viral ID is central to clinical practice.
There are 4 big reasons for that. The first is therapy justification. Modern antivirals are expensive, and some have serious side effects.
So you need to know exactly what you're treating.
Exactly. You need to identify the virus to make sure the patient gets the single appropriate targeted therapy. You wouldn't treat a common cold with a powerful HIV drug.
The second reason is simply that the technology now allows it.
Absolutely. The availability of rapid, novel molecular tech means we can get an answer in hours, not days or weeks, so it's actually useful in the clinic.
Third, it's essential for managing chronic disease.
Think about human immunodeficiency virus, or HIV, or hepatitis C. Accurate diagnosis, and just as importantly, continuous viral load tracking are integral to long-term management.
And finally, the public health mandate.
Specific rapid diagnosis is the entire backbone of public health. It lets us track outbreaks, epidemics, pandemics. It guides everything from hospital resources to vaccine strategy.
So let's review the methods, starting with the original approach, viral culture.
Viral culture is where we grow the virus in cells and watch for the damage, the cytopathic effect. It's undeniably slow and expensive.
But it's not obsolete.
No, because it remains the only method that gives you a viable, living isolate. If researchers need a live sample for further testing, like for resistance or vaccine development, you still need to culture it.
Next up is antigen detection.
Antigen detection looks for the viral proteins themselves. It uses specific tagged antibodies, often with a fluorescent label, to find viral proteins in a sample. An ELISA, or enzyme-linked immunosorbent assay, is a common example.
What are the pros and cons there?
It's much faster than culture, for sure. But its major limitation is that it doesn't work well for viruses with high antigenic heterogeneity.
Meaning viruses that change their surface proteins all the time, like rhinoviruses.
Exactly. If the protein changes, the antibody might not stick anymore.
Okay, moving on to serology, which looks at the patient's immune response.
Right. Serology detects virus-specific antibodies. It's essential for figuring out the stage of an infection. We measure 2 key types.
IgM and IgG.
Right. High levels of immunoglobulin M, or IgM, usually indicate an acute disease, the body's first rapid response.
And the long-term marker.
That's immunoglobulin G, or IgG. IgG levels indicate a past infection or acquired immunity. By measuring both, you can reliably tell if an infection is current and active or if it's resolved. It's vital for diagnosing things like mono.
And finally, we get to what the source is called gold standard nucleic acid detection. This is PCR.
Polymerase chain reaction, or PCR. It revolutionized virology. It's a technique to amplify and specifically detect tiny amounts of viral genetic material. If you know a little piece of the genome, PCR can find it and copy it billions of times. It's incredibly sensitive and specific.
And the evolution of that is quantitative, real-time, reverse transcription PCR. What extra data does that give us?
Okay, so the reverse transcription part is because many important viruses, like influenza, have RNA genomes. So we have to convert that RNA into DNA first.
Right.
The quantitative real-time part means we watch the amplification as it happens. This lets us measure the original amount of viral RNA in the sample, which gives us the crucial figure of viral load.
And that's indispensable for tracking treatment for HIV or hepatitis C.
Indispensable. And it helps with strain identification during outbreaks.
And for complex cases, we have multiplex PCRs.
Multiplexing is just efficiency personified. It lets you test for a whole panel of different viruses, or even viruses and bacteria, all from a single sample at the same time. For something like pneumonia, it can give you a complete diagnostic map very, very quickly.
And you can run these tests on almost anything, right? Respiratory swabs, blood.
Respiratory, cutaneous, blood, you name it. It's a truly adaptable set of tools at the core of modern medicine.
So now we move from the map to the territory itself, focusing on respiratory viruses. And these are, they're the most common cause of illness in humans, but the sources have a shocking statistic. They account for about 30% of child deaths in the developing world.
It completely reframes how you think about a common cold.
It really does.
Let's start with the main culprit for those colds. Rhinoviruses, or HRV. These are small, non-enveloped sRNA viruses. They cause about half of all upper respiratory tract infections.
And how do they get into our cells?
Most of them use a receptor called intercellular adhesion molecule 1, or ICAM1. A few others use the low-density lipoprotein receptor, LDLR. But it's worth noting there's a newly identified species, HRVC, and we actually still don't know its entry mechanism. It just highlights that there are still gaps in our knowledge.
And while the symptoms are usually mild, the pathology is not benign, especially for vulnerable patients.
That is the key clinical detail. HRV disrupts the epithelial cell barrier in our airways. And this disruption, it sets the stage for severe complications.
It opens the door.
It opens the door for bacteria to transmigrate through that broken barrier, and it ramps up inflammation. That's why rhinoviruses are notorious for exacerbating conditions like COPD, asthma, and cystic fibrosis.
Next, coronaviruses, HCoV.
These are enveloped positive strand ssRNA viruses. They cause about 15% of common colds. Historically, we tracked a couple of main groups, but the 21st century brought new challenges, including SARS-CoV in 2003.
And the insight on immunity here is a bit concerning.
It is. There are no effective vaccines for these common strains, and any immunity you get from a natural infection is fleeting. It wanes significantly after just a year or two.
So you can just keep getting them.
Reinfection is common throughout your life.
Okay, let's move to adenoviruses, HADV. These are dsDNA viruses.
Right, and they cause a whole spectrum of illnesses. Acute respiratory disease, epidemic keratoconjunctivitis, pink eye outbreak.
Laryngitis, things like that.
Exactly. And the sources really emphasize their diversity. There are over 67 different serotypes known to infect humans, and they are constantly evolving through homologous recombination.
Which makes vaccination a huge challenge.
A huge ongoing challenge. And their effects on cells are interesting. In epithelial cells, they replicate until the cell bursts cell lysis. But in lymphoid cells, fewer particles are released, and the cell can survive, potentially setting up a persistent infection.
And crucially, there is an oncogenic link with adenoviruses.
There absolutely is. They can cause malignant transformation in a couple of ways. First, their viral proteins, E1A and E1B, can directly mess with the host cell's growth regulation.
And the second way?
Second, the adenoviral DNA can physically integrate itself into the host cell's DNA, which is a high-stakes event that can drive cancer.
Now for the global heavyweight. Influenza. Enveloped, negative-sense ssRNA, and a segmented genome.
And that segmentation is critical. Influenza causes seasonal infections, which are bad enough, but it's also responsible for global pandemics. The complications are severe: pneumonia, hemorrhagic bronchitis, and croup.
And certain patients are hyper-vulnerable.
Yes. Patients with comorbidities, like chronic pulmonary or cardiac disease or diabetes, they face much, much higher risk.
Might classify them into A, B, and C.
Type A is the most challenging because it's zoonotic. It infects a wide range of species, like birds and pigs, creating these huge reservoirs for the virus.
Whereas B and C are mostly just in humans.
Almost exclusively, yes.
And the specific subtyping relies on those surface keys: hemagglutinin H and neuraminidase N.
Correct. And the sheer number of variants, 16 for H, 9 for N, means the virus has this enormous capacity for generating new combinations.
Which brings us to the crucial difference between antigenic drift and antigenic shift. This is a distinction every professional needs to master.
It is. Antigenic drift is the slow, continuous process. Think of it like constant refinement. It's the acquisition of small, little point mutations in the H and N genes.
Just tiny changes.
Tiny changes. But they're enough to alter the surface profile so the virus can escape the immunity you built up from past infections or vaccines. This is why you need a new flu shot every single year.
Antigenic shift, on the other hand, is the catastrophe.
Shift is the major, abrupt change. And it only happens in type A influenza because of that segmented genome and its zoonotic nature.
So you need two different strains infecting the same host.
Exactly. When, say, an avian flu and a human flu co-infect the same host, often a pig, which acts as a mixing jessel, their RNA segments can get swapped around, recombined.
And you get a brand new subtype.
You get an entirely new subtype, like H5N1, that the human population has zero immunity against. And that's how you get a pandemic.
Okay, finally in this section, the paramyxoviridae family. enveloped negative strand ssRNA viruses. Let's start with respiratory syncytial virus, RSV.
RSV is the most common respiratory tract infection, full stop. It can cause severe complications like bronchiolitis in 1 to 2% of cases, making it a huge threat to infants and the elderly.
And the lack of a licensed vaccine is a major clinical frustration.
It is. We have monoclonal antibodies like palivizumab, but their effects are consistently reported as being, at best, pretty mild.
Next, parainfluenza viruses, HPIV.
Second most common cause of child hospitalization for respiratory illness, right after RSV. Certain subtypes cause croup with that classic barking cough. Others cause bronchiolitis and pneumonia. And rarely it's been linked to severe neurological issues like viral meningitis.
Now for the classic ones, mumps.
Mumps is highly infectious, spread by saliva. The hallmark is the swollen salivary glands, the parotids, but the complications go way beyond that.
Right, it can attack other glandular tissues.
Exactly. Oophouritis, orchitis, pancreatitis, viral meningitis. And hearing loss is a commonly overlooked consequence. Transient hearing loss happens in about 1 in 20 cases.
And permanent loss.
Permanent loss is rare, about 1 in 20,000, but tragic when it occurs. The good news is clearance gives you lifelong immunity, and the MMR vaccine is very effective.
And the most dangerous of this family, measles.
Measles is, well, it's terrifyingly contagious, spread by respiratory droplets. The classic symptoms are the coryza, conjunctivitis, fever, and then the rash. But crucially, you're infectious for days before the rash even appears.
The fatality risk is significant, especially for infants.
Severe complications include encephalitis and pneumonia, with a fatality risk of about 1 in 5,000. And it can cause permanent bilateral deafness in about 1 in 1,000 cases. Again, the MMR vaccine provides highly effective, lifelong protection. It's a paramount public health tool.
Now we're moving into viruses that establish these chronic relationships with the host, starting with the herpes viruses, the HHVs.
Right. These are enveloped dsDNA viruses, and they are a very common cause of oral mucosal infections.
And the unifying characteristic of the whole family is latency.
Latency, exactly. Take herpes simplex virus, HSV1 and AGST2. HSV1 gives you the common cold sores. The immune system clears the active infection just fine.
But the virus never actually leaves.
It never leaves. It enters a chronic latent phase, hiding silently in the cell bodies of the nervous system, in the sensory ganglia. Our current antivirals can treat an active outbreak, but they are completely useless against this latent reservoir.
Which is why it can keep coming back.
Recurrence is a certainty.
Epstein-Barr virus, or EBV, is similar. It's the cause of mono.
Yes, or glandular fever. And its entry is highly precise. It uses the CD21 receptor to get into B cells and the beta-1 integrin receptor to get into epithelial cells. And just like HSV, it establishes a chronic latent phase.
And that latency is the critical link to several serious cancers we'll get to later.
Precisely. Hiding from the immune system like that makes it a formidable long-term threat.
Let's briefly mention retroviruses.
Retroviruses are defined by their unique enzyme, reverse transcriptase. They are ssRNA viruses, and they use that enzyme to make a dsDNA copy of their RNA genome. That DNA copy can then integrate into the host's own genome.
And the classic example is HIV.
The human immunodeficiency virus, yes.
Next up, ubivirus, which causes rubella, or German measles.
Rubella is a positive-sense ssRNA enveloped virus. Its key feature is that it's a purely human virus. There's no animal reservoir.
Which means it's theoretically eradicable.
With global vaccination, yes. But it's so feared for one reason. In kids, it's often mild, but if a non-immune woman contracts it during pregnancy,
it's devastating for the fetus.
It can cross the placenta and cause congenital rubella syndrome, leading to profound and permanent damage ocular defects, neurological problems, and, crucially, deafness.
And finally, let's analyze human papillomavirus, or HPV, a family of DNA viruses with over 70 subtypes. The clinical table associating types with pathologies is essential here.
It is. You have to distinguish between low-risk and high-risk types. For benign things like common warts, you're looking at types 2, 7, 22. For certain oral lesions, other types.
But the real clinical concern is malignancy in the head and neck.
Exactly. For laryngeal papillomatosis, the most common culprits are type 6 and 11. For oropharyngeal carcinoma, types 16 and 18 are the high-risk ones we look for. This specificity is why viral typing is crucial for prognosis and treatment.
Now we can execute that comprehensive clinical review detailing how these viruses show up in head and neck pathologies. Let's start with that laryngeal papillomatosis.
Right, so this is predominantly associated with HPV-6 and HPV-11. And because these papillomas, these growths, are in the larynx, the primary concern is progressive airway obstruction, especially in children.
And the treatment is just continuous intervention.
It is. It requires repeated surgical ablation, often with laryngoscopy, just to keep the airway open. Malignant transformation is rare, thankfully, but it can happen, usually tied to risk factors like smoking.
Let's quickly group the viral involvement in other common conditions. First, middle ear infections and chronic respiratory issues.
Acute otitis media, or AOM, is linked to a whole host of common respiratory agents. Parainfluenza, influenza, rhinovirus, RSV, adenovirus.
The usual suspect.
The usual suspects. And it's the same list for viral exacerbations of things like asthma and COPD. It just reinforces that a simple cold can rapidly destabilize a patient with underlying lung disease.
We also see extensive viral involvement in severe neurological issues like encephalitis and viral meningitis.
The list of causes for encephalitis is long and includes common viruses like HSV, VZV, mumps, measles. And for viral meningitis, it's a similar story. The sheer diversity here is why immediate, precise diagnostic testing with those PCR panels we talked about is so critical in an emergency.
And for more routine issues like pharyngitis, tonsillitis,
Adenovirus, EBV, HSV, they account for a huge percentage of non-bacterial sore throats.
Okay, now let's tackle the medical mystery surrounding Bell's palsy.
Bell's palsy is an acute temporary paralysis of the facial nerve. It's idiopathic, meaning we often don't know the cause, and it's generally benign. About 71% of people get complete resolution, even with no treatment.
For decades, the dominant theory has been viral reactivation.
Right, the idea that a latent virus, usually herpes simplex virus type 1, reactivates and causes inflammation and swelling of the facial nerve.
And since that nerve runs through a narrow bony canal, the swelling entraps it.
Yeah, exactly. And this theory was heavily supported when researchers found the HSV-1 genome in the endoneurial fluid surrounding the nerve in some patients.
But here's where we have to be critical thinkers. The clinical outcome data seems to flatly contradict this.
This is the critical professional insight. A huge, authoritative Cochrane review looked at trials using anti-HSV antivirals for Bell's palsy and found something startling.
What was it?
The antivirals were no more effective than a placebo in achieving complete recovery.
So even if the virus is there, treating it doesn't help the patient get better.
Precisely. This massive contradiction suggests that either HSV-1 is just an incidental finding, or its presence is a secondary effect, not the primary cause of the paralysis. We have to question that long-standing assumption.
Let's shift focus to sensorineural hearing loss, SNHL, which has a wide array of viral culprits.
Right, and we have to distinguish between congenital, so present at birth, and sudden SNHL. For congenital SNHL, cytomegalovirus, or CMV, is the undisputed champion.
It's the most common non-hereditary cause.
It is, and the progression rate is alarming. Hearing loss was found in a third of symptomatic infants and a fifth of asymptomatic infants with congenital CMV. And it's often not static. It can progress or fluctuate over time.
And how does CMV actually damage the delicate inner ear?
A histological study showed that CMV infects the structures involved in endolymph secretion and potassium homeostasis.
So it messes with the fluid and the chemical balance.
Exactly. The inner ear needs a perfect balance of potassium-rich fluid for the sensory cells to work. The hypothesis is that CMV damage messes up that potassium balance, which causes a secondary degeneration of the sensory structures in the cochlea. It basically short-circuits the hearing mechanism.
What about idiopathic sudden SNHL, the unexplained acquired kind?
There are three potential viral mechanisms. First is direct invasion, where the virus gets into the cochlea and causes damage. Second is latent virus reactivation within the inner ear tissues, similar to the Bell's palsy theory.
And the third is more indirect.
The third is that a systemic viral infection triggers an antibody response that then accidentally cross-reacts with an antigen in the inner ear, or it triggers some other circulating molecule that causes cellular stress. Either way, the damage is indirect.
Finally, let's detail the pathogenesis of acute otitis media, AOM, which almost always follows a viral upper respiratory tract infection.
The link is profound. Data shows a third of kids with a viral URTI will develop AOM within 4 weeks. The virus plays a pivotal enabling role.
So how does a cold set the stage for a bacterial ear infection?
It's a chain reaction. The virus causes nasopharyngeal inflammation. This alters the mucus, makes it thicker, and reduces mucociliary clearance, the little hairs that sweep things out.
So everything gets stuck.
Everything gets stuck. This leads to eustachian tube dysfunction, creating a stagnant, negative pressure environment where bacteria can thrive and invade the middle ear.
So the virus doesn't cause the AOM itself, but it creates the perfect environment for bacteria to take over.
Exactly. Rhinovirus and RSV are the most common culprits. It's fundamentally a viral bacterial interaction.
We now confront the most serious dimension of viral biology, oncoviruses, viruses that cause cancer. And the scale of this is immense. In 2008, nearly 2 million cancer cases worldwide were attributed to infections.
Right. And for head and neck cancer specifically, the International Agency for Research on Cancer, IIRC, has definitively identified 2 causative agents, Epstein-Barr virus, EBV, for nasopharyngeal cancer, and HPV for oropharyngeal cancer.
Let's focus on EBV, the first human oncovirus identified. It's implicated in several cancers, starting with Burkitt lymphoma.
Burkitt lymphoma is a B-cell tumor, often in the jaw area of young children. EBV is detectable in 95% of these tumors. But in endemic areas, malaria is considered a crucial cofactor.
So the 2 infections work together.
They interact to significantly raise the risk, yes.
EBV is also central to nasopharyngeal carcinoma, or NPC.
NPC has a very high incidence in specific populations, mainly in South China and Southeast Asia. This clustering suggests cofactors are at play, like genetics and environmental factors like diet.
But the viral link itself is solid.
Oh, absolutely. The latent EBV genome is consistently detected in virtually all endemic cases, and patients have much higher antibody titers. The causal link is widely accepted.
EBV also causes a unique premalignant condition in the immunocompromised called hairy oral leukoplakia.
Right, these white, shaggy lesions on the side of the tongue. It occurs almost exclusively in immunosuppressed people, especially those with HIV. Here, EBV is actively replicating. Antivirals can make the lesions regress, but only transiently.
And the final link is to other lymphomas.
In immunocompromised individuals, the immune system can lose control over a persistent EBV infection. This leads to the uncontrolled proliferation of virus-carrying B cells, and that can lead directly to lymphomas like Hodgkin's.
Now, for human papillomavirus HPV, we know HPV-16 is causally linked to oropharyngeal cancer, especially tonsil cancer.
Yes, the virus integrates its DNA, and its viral proteins interfere with crucial tumor suppressors like p53, driving unchecked cell growth.
But here is the critical prognostic insight that dramatically impacts treatment.
This is a major detail every clinician must know. Research has shown definitively that HPV-positive head and neck cancer is associated with an improved prognosis compared to HPV-negative tumors.
Which means you can treat it less aggressively.
Often, yes. The patient might be able to tolerate a de-escalated chemotherapy or radiation regimen and still have excellent survival rates. Knowing the HPV status is essential for treatment planning.
Having mastered the threat, we now master the arsenal. Prevention starts with vaccines, which are highly effective for diseases like measles and mumps, but struggle against highly diverse viruses like rhinovirus.
Right, and there are 3 main strategies. First, live attenuated vaccines, like the MMR. These use a weakened live virus that can't cause disease but still triggers a robust, lifelong immune response.
Second, inactivated viruses.
Like for polio or hepatitis A, the virus is rendered unable to replicate, it's dead, but the immune system can still recognize it and generate protection.
And third, modern subunit vaccines, like the one for HPV.
These are the safest because they only contain isolated pieces of the virus, specific proteins. There's no genetic material at all, but they still elicit a protective response.
Now let's turn to antivirals. And we have to reiterate, these drugs do not kill the virus, they just impede a specific function.
Which demands high specificity. Let's start with an older category for influenza. The M2 ion channel blockers, like amantadine.
Okay.
These are used extensively, but resistance emerged rapidly. They don't work on influenza B, and the side effects aren't great. The clinical guidance now is clear. They are generally not recommended.
Next, interferon, or IFN.
Interferons are naturally occurring signaling proteins with potent antiviral effects. They have direct cellular effects, and they also stimulate host anti-tumor responses.
But their clinical use has been surprisingly limited.
That's right. They've proven much less effective in vivo than they were in vitro, and their use is restricted by severe adverse effects like major flu-like symptoms. They're still used successfully in combination therapy for chronic hepatitis B and C, though.
The gold standard for herpes infections is acyclovir, and its specificity is remarkable.
It's a phenomenal example of drug design. Acyclovir is a prodrug. It's inactive until it encounters the herpes virus's own enzyme, thymidine kinase.
So the virus has to activate the drug that will stop it.
Exactly. The virus's own enzyme activates it, and then it inhibits herpes DNA replication. Since our cells don't generally have that viral enzyme, the drug is highly specific with low toxicity.
And it's used for acute treatment and also for prevention.
Yes, especially prophylactically in high-risk patients like organ transplant recipients and in high doses for severe infections like encephalitis.
A related but more toxic drug is ganciclovir.
Ganciclovir has better activity against cytomegalovirus, or CMV. But because it's more toxic, it's strictly reserved for severe CMV conditions in immunocompromised patients, like CMV retinitis.
Okay, now the modern class for influenza, neuraminidase inhibitors. Zanamivir, oseltamivir.
These target the neuraminidase protein, the N spike. By blocking it, they prevent new viruses from being released from the cell surface. It limits the spread from cell to cell.
But the clinical efficacy data here is a major point of discussion. The Cochrane review has some sobering insights.
It did. The real-world benefit is modest. The review found that in otherwise healthy adults, these drugs shorten the duration of symptoms by less than a day.
Less than a day. And it didn't reduce hospitalizations.
Correct. So the best clinical practice guidance is to restrict their use. They're recommended for prophylaxis only in at-risk adults who weren't protected by the vaccine, and for treatment only for those at-risk patients when we know flu is circulating.
Our final antiviral is ribavirin.
Ribavirin has impressive in vitro activity against a huge range of viruses, but its in vivo use is severely limited by dose-limiting toxic effects, especially hemolytic anemia.
So where is it used?
As part of combination therapy for hepatitis C, and it's also given by aerosol for severe pediatric RSV, though its efficacy there remains controversial.
Let's consolidate the summary of best clinical practice from all this.
The directives are clear. 1, prioritize flu immunization for all at-risk groups. 2, strictly restrict neuraminidase inhibitors to those same at-risk populations. 3, use acyclovir immediately and in high doses for severe herpes infections. And 4, the critical negative finding. Anti-HSV antivirals offer no significant benefit for Bell's palsy.
The future of virology is perhaps the most exciting part of this. We're moving beyond just blocking viruses to actually using them as tools. This is virotherapy.
Right, and it's been re-energized by genetic engineering. The most promising area is oncolytic viruses engineered to specifically target and kill cancer cells while leaving healthy cells alone.
The sources mention a natural cancer-targeting virus already in advanced trials.
That's the Rio virus. It's in phase 3 trials for head and neck cancer, used along with chemotherapy. The idea of a virus as a targeted cancer weapon is a huge paradigm shift.
And the other side of this is gene therapy.
Using viruses as delivery vehicles to transport therapeutic genes. But we have to be cautious. Remember the risk of inadvertent insertional mutagenesis, where the virus integrates in the wrong place and causes a new cancer.
A risk realized in some early trials.
Tragically, yes. But momentum continues. A treatment called Gendicide is licensed for head and neck cancers in China.
Another exciting avenue is bacteriophages, viruses that infect bacteria.
In the face of growing antibiotic resistance, these offer a critical potential alternative. They are precise, self-replicating agents that kill pathogenic bacteria. They could be a huge part of future antibacterial treatment.
And what about new drugs? Novel small molecule antivirals.
Research is focused on finding compounds that interfere with highly specific viral processes, like jamming the influenza polymerase complex. But the more specific the drug, the more you need ultra-precise diagnostics to know when to use it.
And finally, ongoing vaccine research.
Yes, clinical trials are underway for polyvalent HPV vaccines, both to treat existing respiratory papillomatosis and to prevent future head and neck cancers. Prevention is, and will always be, the most powerful tool.
This deep dive has taken us from the most basic acellular structure all the way to the frontiers of genetic medicine. We've really mastered these foundational concepts. Positive versus negative sense genomes, antigenic shift, the life cycle steps that antivirals exploit.
We saw how PCR-driven diagnostics are now the indispensable foundation of clinical practice. And we detailed the extensive connections between common viruses and head and neck pathologies, from the viral priming needed for AOM to the complex mechanism of CMV-induced hearing loss.
We also established that the battle is nuanced. Vaccination is powerful, but antivirals must be used with discipline, from the high reliability of acyclovir to the limited benefit of drugs like oseltamivir. And we highlighted that critical contradiction in Bell's palsy, forcing us to constantly question accepted ideas.
And we confirmed that EBV and HPV are primary oncoviruses in the head and neck, but with that counterintuitive insight. HPV-positive cancer carries an improved prognosis, which changes the whole treatment landscape.
Right. And our future strategies are perhaps the most intellectually challenging. We often see viruses solely as adversaries, as plagues to be wiped out. But with virotherapy and bacteriophages, we're learning to leverage their inherent precision. We're turning them from threats into tools.
Which leads to our final provocative thought for you, the learner. As we develop the capability to engineer viruses with precise genetic payloads, how might our clinical approach fundamentally change when these entities are seen less as a plague to be eradicated and more as incredibly complex, customizable medical delivery systems for things like targeted cancer destruction or gene editing? That transformation is happening right now.